Hello, dear students. We are starting a new chapter today that's molecular base of inheritance. Revision shorts we are doing, we'll be talking about DNA. DNA is the blueprint of life. Every organism, from bacteria to humans, stores its genetic information in the form of DNA. DNA is long polymer made up of repeating units, which are called as deoxyribonucleotides. The length of DNA varies from organism to organism and is expressed as a number of base pairs. So when we talk about this bacteriophage, this phage, the length is around 5386 nucleotides. 5386 nucleotides. Then when we talk about lambda phage, 48,502 base pairs. Then you have E. coli 4.6. This is important. 4.6 into 10 to the power 6 base pairs. And this one is 48 and then 502 base pairs. This is the data from your textbook. You can check it on the textbook also. When we talk about human beings, what haploid DNA? It is 3.3 into 10 to the power 9 base pairs. This is haploid one strand of the DNA. Then structure of the polynucleotide. In the nucleotide you have a base, you have sugar, and you have phosphate, pentose phosphate. So this is the composition of these are the components of nucleotide. When we talk about nucleoside, you don't have phosphate in the nucleoside. Then nitrogenous base. In nitrogenous bases you have like purines. Purines Purines are two, that is adenine and guanine, and then you have pyrimidines, PY. You have cytosine, thymine, and uracil in case of RNA. So in DNA you have bases ATGC, that is adenine, thymine, guanine, cytosine. RNA you have uracil, which is replacing the thymine. Now sugars in DNA you have this deoxyribose sugar. In RNA you have ribose sugar. That means that in ribose sugar, you have extra OH group at 2 position. Now, formation of nucleotide, you have base plus sugar plus nucleoside. Base plus sugar, you have nucleoside, and then nucleoside plus phosphate is nucleotide. So, nucleotides, they join by 3' 5' phosphodiester linkage. These nucleotides, which we are talking about when they form the DNA, they join from 3', 5' phosphor. They form phosphodiester linkage. Then one end of this nucleotide has free phosphate, 5' end. 5' end, you can see here in this diagram, 5' end is having free phosphate, or this one, free phosphate, and 3' end is having hydroxyl group, OH group. So this gives DNA directionality, 5', 3' directionality. So now, when was this DNA discovered? How was the structure revealed? So DNA was first identified by Frederick Miescher and named as a nuclein. He named it nuclein. Structure remained unknown due to technical limitations. In 1953, Watson and Crick proposed double helical model based on, and this is important thing. This double helical model was based on X-ray diffraction data, which was given by Rosalind Franklin. It's very important, X-ray diffraction data. So work of Maurice Wilkins also helps us here. Then we have one rule that's called Chargaff's rule. In Chargaff's rule for double-stranded DNA, this applies to double-stranded DNA. Please remember, adenine is equal to thymine and guanine is equal to cytosine. This is what Chargaff's rule say. So adenine, how much is adenine? Same amount will be of thymine in DNA, and how much is guanine? Same amount will be of cytosine. This is not bonding here. I'm not talking about double bond here. This is equal to adenine amount is equal to thymine amount, and guanine amount is equal to cytosine amount. Please remember this thing. This is very important rule. Questions are coming on this. Now, double helical model. What are the key features? DNA has two polynucleotide chains. There are two polynucleotide chains in the DNA. Sugar phosphate backbone is outside, base project inside. Chains are antiparallel, 5', 3' to 3', 5'. So it's been shown here. It's a double-stranded polynucleotide chain. Then base pairing, adenine pairs with thymine. You can see here, adenine thymine is a double hydrogen bond. And guanine pairs with cytosine, it's a triple hydrogen bond. Always purine pairs with pyrimidine, maintains uniform distance. DNA is a right-handed helix. This is also an important point. The DNA is right-handed helix. One turn is equal to 10 base pairs. One turn has 10 base pairs. Pitch is 3.4 nanometers. The distance between bases is 0.34 nanometers. So base staking plus hydrogen bonds give stability to the DNA. So remember, base staking and hydrogen bonds, they give stability to the DNA. Now, why distance remains constant between these purines and pyrimidines? You know, purines are large, always pair with pyrimidine, which are small. Purine, if there is purine interaction, it will be too wide. If there is pyrimidine interaction, then it will be too narrow. Hence, uniform diameter of DNA is there when it is pairing with pyrimidine. Now, complementarity and genetic significance of this. If one strand sequence is known, the other sequence can be predicted. If you have two strands of the DNA, the sequence of one strand is known, you can easily find out the sequence of the other strand because it's complementary. Each strand acts as a template. DNA replication becomes accurate. Daughter DNA molecules are identical to parent DNA. So this is all about this. This is the polynucleotide over here, which is given in your textbook. It is showing the polynucleotide. Like you have cytosine, it's a nitrogenous base. Then you have sugar, and then you have phosphate. So this is one nucleotide. This is another nucleotide. This is another nucleotide. There are many nucleotides form one chain of the DNA itself or RNA itself. So these chains, they are connected and forming the polynucleotide. And these are hydrogen bonds. Please remember. And these bonds which are here, these are phosphodiester bonds. Now, there's one more thing that's given in the introduction is central dogma. This was proposed, this was given by Crick. So remember it. It means from DNA, transcription happens, mRNA is formed, and from mRNA, protein is formed. This whole process pattern is called as central dogma. From DNA, transcription happens, mRNA is formed. From mRNA, protein is formed. Translation happens, protein is formed. DNA undergoes replication also. That's why this thing has been shown here. So this, well, if somehow this RNA forms DNA, you may have seen in HIV virus. It is possible because of one of the enzymes that's called a reverse transcriptase, and this is called a reverse transcription. So remember all these points; they are very important. I hope you understood this structure of the DNA, and it will help you in exam. So you can watch this video 2-3 times so that you can understand it properly. Thank you.